Thank you for deciding to stay here and not going to the lunch. And I thank organizers for an opportunity to give a talk about the studies we do in the KTH. So our group is particularly interested in nanocelluloses and in wood-based materials. And in this study about the nanocelluloses, I would first explain why we are so interested in this. Nanocelluloses are relatively new nanomaterials which have, from our point of view, a great promise for different applications. First of all, because of their great mechanical properties. The second, probably most important, that it comes from abundant, renewable resource, probably the most abundant on Earth. It comes from wood, plants, algae, even marine animals, very few of them. So at the same time, it is biocompatible, it's biodegradable, and it's possible to, via quite straightforward chemical functionalization, to even adjust the properties to the application of your choice. So we are very fascinated by this material. And what are the nanocelluloses? Nanocelluloses could be of two types, the fibrils or the nanofibrils or nanocrystals. The nanofibrils are quite prolonged compared to the nanocrystals, which are much shorter. These are obtained from the fibrils aggregates. The cellulose nanofibrils could be defibrillated from them, while the cellulose nanocrystals are much shorter and obtained through the chemical procedure of hydrolysis. If you study these nanomaterials with a conventional bulk vibrational spectroscopy methods or NMR spectroscopy, you typically have two signals, the crystalline signal, the signals of crystalline cellulose and amorphous cellulose. And it's not clear where they are from. And there is a textbook explanation that the small fibrils may contain alternating regions of the highly ordered crystalline cellulose and less ordered or disordered amorphous cellulose. And these signals come from them. So here's what the answer we want to, here's the question we want to answer, I'm sorry. That if these regions exist, interestingly, for example, the cellulose nanocrystals, which are much shorter, supposed that during the hydrolysis, these amorphous regions are destroyed and you get the shorter crystals. So, obviously, conventional spectroscopy techniques lack of the spatial resolution, and that's the basically natural choice of us was the nano-FTIR spectroscopy, which combines both great spatial resolution and possibility to characterize the chemical samples with the obtain chemical information. And before we go to the nanocellulosic materials, we first, as a preliminary step, we wanted to obtain signals which represent both the crystalline and amorphous cellulose. For this, we obtained two freestanding films, basically very thick, up to 1 millimeter thick film. And we acquired the conventional ATR-FTIR spectra of those and the nano-FTIR and compared them. You can see that the peak positions in the highly crystalline and highly amorphous samples are similar, as well as the profiles, spectral profiles. This actually inspired us a lot to continue with the nanocellulosic materials because it shows that the nano-FTIR is able to identify either of the highly crystalline and amorphous material. So the first nanocellulosic sample was cellulose nanocrystals made of cotton. This is a plant which produces highly pure cellulose, and those have the typical height of 3 to 10 nanometers only. With our neaSNOM setup, we were able to very carefully study the single crystals in multiple points. And one of the hypotheses we wanted to check is that if the endpoints where the crystal ends has more of amorphous cellulose compared to the main body of the crystal. We obtained the nano-FTIR spectra, and you can see that the signal from the endpoints didn't very much compared to the spectra obtained from the main body of the crystal. It's all highly crystalline cellulose based on the peaks. That was interesting, and we decided to move to the next cellulosic nanomaterial, the cellulose nanofibrils made of the tunicate. Tunicate is a marine animal which also produces the highly pure cellulose. So the tunicate cellulose nanofibrils are a bit smaller. They are 3 to 6 nanometer high only, but again, the difference between the crystals is that they are much, much longer. First, we again studied the main body, the straight regions of the fibrils, obtaining the spectra. These were highly crystalline cellulose, indicating highly crystalline cellulose, and compared to the ends of the fibrils, here, 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 those also show the spectra of the highly crystalline cellulose as with the crystals. The next areas where we expected to see the amorphous regions are the so-called kinks. Those do not present in the crystals because those are much shorter. The kinks are the areas where the fibrils suddenly change its direction. Carefully studying them, however, we did not observe the increase of the amorphous signals. The peaks were indicate the highly crystalline cellulose there. And another morphological feature which present in the fibrils are the twists. Those are the areas where the cellulose fibril rotates around its main direction. These areas are quite abrupt because of the drying on a surface. In our case, all our samples were put on the silicon wafer surfaces. So we studied them also very carefully, again, observing only the signals of the crystalline cellulose. However, interestingly, we at the same time observed that in the middle of the twists, we lose some of the signal. This puzzled us a lot. We asked the neaSpec, Adrian specifically, and he kindly agreed to help us. He measured, we wanted to know what happens. Is it just our sloppy work, or is there a reason? So he obtained for us, or the neaSpec team, 2 images, one with the scattering nSNOM image and another one using the new technique called tapping AFM-IR+. Though these techniques have different principles, and the images were acquired at the wave number of the highest crystalline cellulose peak absorption. We are interested in the absorption images, and you can see that not only in nano-FTIR, but also in those images, the signal in the middle of the twist area decreases. After some discussions, we decided that it possibly happens, just as the previous speaker described, that due to rotation of the crystal planes of the cellulose fibril, in the middle of the twist, it happens that they become an in-plane. They're arranged in plane perpendicular to the direction of the AFM tip, which leads to the decrease of the observed decrease of the signal, which I think was quite interesting part of the study. The next cellulose material was cellulose nanofibrils made of wood, and those are much smaller than the previous, both crystals and fibrils, only 1 to 3 nanometers high. And those were so tiny that I was not able to obtain a reproducible or repeatable spectra from the single fibrils. Those spectra which I was able to obtain and show you, those again contain crystalline cellulose. Those were obtained from the aggregates and as well, not aggregates, sorry, from the overlap of the fibrils. Also, we studied the CNF aggregates, you can see on the optical microscopy image here, as well as the carboxymethylated fibrils. Those are the originate material that fibrils originate from, and the cellulose fibers before the tempo oxidation. However, the spectra which I acquired are, again, show mostly the crystalline cellulose. And of course, with the decreasing of the volume of the probing volume, the signal decreases, but we still see the highly crystalline cellulose. And on this, I want to finish with the results. Yeah, thank you. What I want to say in the very end that neaSNOM setup is highly capable. We were able to obtain reproducible signals from such small materials as cellulose nanofibrils and crystals, which could be up only 5 nanometer high and 10 nanometer wide. Regarding the nanocelluloses, we obtained the peaks. We observed the peaks of the crystalline cellulose in all the studied points, both from the main parts of the samples and from the morphological features we studied, which indicated that the textbook explanation on the alternating domains of the amorphous and crystalline cellulose seems to be outdated. And the main conclusion of this outcome is that the amorphous signal observed by the conventional spectroscopies should come most likely from the molecules on the surfaces of the cellulose nanofibrils and nanocrystals. With this, I would like to thank my colleagues from the KTH, Research Institutes of Sweden, and of course, Adrian from the neaSPEC and Swedish funding organizations without whom this project was not able to be performed. And of course, you for your attention.